All right, everybody, take your seats. Roberto, take your seat. Personally. All right, so as people are settling in, I hope you've enjoyed the first session. I think this first session was spectacular. I want to thank all the speakers and the debaters, and I would say that the people who won from the debates was the audience, because I know I learned a ton, and I think this is really providing excellent material for all of you to discuss in your breakout sessions, sort of the innuendo and thinking about how to use these drugs. All right, so in Plenary 2, we have a really nice series looking at each of the disease groups we're going to work with. Then we will have a 15-minute question and answer for the speakers, so if you'll remain up here on stage after your talk, that'd be great. And then we end before lunch with possibly the most important panel that we have today, which will be our patient panel. And so very exciting Plenary Session 2. Our first speaker is Nicola Tomas, and I was with him last week at the Podocyte meeting, and he's going to talk about antigen-specific B cell targeting for membranous nephropathy, which is some of his really cool work. So take it away, please.
Thank you very much. Thank you so much for setting up this fantastic meeting and inviting me and giving me the chance to present some of our work and also others' work, actually. So these are my disclosures. So antigen-specific B cell targeting. So how could we do that, and why should we do that? And can we maybe consider this as something like the holy grail of B cell depletion? So you all know how it works. Normally, we have a B cell, it expresses certain surface markers, such as CD20. We give a monoclonal antibody binding there, and we get activation of effector cells, such as NK cells, could also be phagocytes, and these eliminate the target cell. CAR T cells, or CAR NK cells, on the other hand, for example, they show a single-chain variable fragment, again binding to the surface target and eliminating the cell, but these cell surface targets are expressed by certain B cell subsets, and you will always target the cells that express this, this molecule, which are not only the autoreactive cells, but basically all cells. And if we want to specifically target the autoreactive B cell, then we make use that these cells express a surface immunoglobulin. And if we succeed in targeting cells through this surface immunoglobulin or B cell receptor, then we may exclusively deplete the autoantibody producing cells. So why membranous nephropathy? So I believe from all the diseases that we are about to discuss during this meeting, in membranous nephropathy we have the clearest causal relationship between an antibody and the disease. So my personal opinion is it's very simple. There's an antibody, it binds to a target antigen on the podocyte and induces disease. There's not a lot of inflammation, not a lot of T cells in the kidney, and so on. And we could actually experimentally show this a couple of years ago for THSC7A antibodies by purifying the THSC7A specific autoantibody fraction, transferring it into mice that then developed something like a membranous nephropathy with granular IgG deposition and proteinuria. Now for PLA2R, this is more difficult because PLA2R is not expressed by mice and rats, which makes these experiments more difficult. But we generated a transgenic mouse line expressing PLA2R in podocytes. And in a recent experiment, we purified the IgG fraction from patients with PLA2R membranous nephropathy, and we injected the anti-PLA2R IgG, comparing it with the serum that was depleted of the antibodies and with a control IgG. And you can see here that these animals develop proteinuria. And if we look at the histology, it looks very beautifully like membranous nephropathy with strong granular IgG, also IgG4 because this is the predominant subtype, and we see complement deposition like C1q, factor B, C3, and C5. So confirming the very clear, very clean relationship between autoantibodies and the development of the disease in membranous nephropathy. So I was asked also to quickly talk about the role of broad B cell depletion in membranous nephropathy. I don't want to touch too deeply on that. We have different types of B cells expressing different surface molecules. We have rituximab, obinutuzumab, ofatumumab, for example, targeting CD20, and we also have compounds targeting plasma cells like bortezomib, daratumumab, and felzartamab. Now, in membranous nephropathy, there are several randomized controlled trials that have proven efficient that rituximab actually depletes B cells and leads to improvement of clinical outcomes, namely the GEM-RETUX trial, the MENTOR trial, the STARMAN trial, and the RECYCLO trial. However, 30 to 40% of complete and 60 to 85% of complete or partial remission rates at 24 months using rituximab, meaning that there is a large number of patients that does not respond to rituximab. However, or nonetheless, rituximab is part of the current guidelines, and a large number of patients should receive rituximab according to the KDIGO guidelines in the moderate and high-risk groups of membranous nephropathy. So there are other compounds like obinutuzumab and ofatumumab which differ in their characteristics, for example, in regard to that they are humanized or fully human IgG1, that they bind to different epitope, that they have different effector functions in regard to complement and antibody-dependent cellular cytotoxicity, as well as antibody-dependent cellular phagocytosis. So there are differences between these antibodies, and it's certainly a matter of debate today and over the next days how we can use these different molecules to improve the therapy here. This is just one example comparing obinutuzumab and rituximab retrospectively for primary membranous nephropathy, and you can see here in the response rates and indicating that maybe obinutuzumab may be more effective for membranous nephropathy. Now, the antigen-specific B cell depletion. This is a graph from a recent review by Dan, who is here in the audience, and this review strongly favors CD19 as a target to deplete B cells for autoimmune diseases because it is more broadly expressed, as you all know, compared with CD20 in green, which has like significant gaps in this B cell development stages. And what I found particularly interesting is that the surface IgG, which you can see here in blue, so the surface IgG receptor, has a very similar expression profile as CD19. So in theory, if we have an effective agent, we may be able to eliminate B cells through this receptor, affecting similar B cell subtypes as CD19 targeting, however, only the ones that produce the autoantibodies. And there are several ways to do that. So this is the conventional CAR T cell strategy, again, with a single-chain variable fragment as part of the CAR targeting a tumor antigen or a B cell antigen for autoimmune disease. And we can reverse this principle by inserting a part of the antigen as part of the CAR, and then it's a CAR with 2A, so the chimeric autoantibody receptor, because it's basically now the receptor for the membrane-expressed autoantibody that is expressed by these autoreactive B cells. And a second option, for example, could be the use of bispecific T-cell engagers, again, like replacing the anti-CD20 or anti-CD19 component of such a bispecific T-cell engager with a part of the antigen that you can see here, which again should lead to elimination of the specific target B cell. So we did this for membranous nephropathy using chimeric autoantibody receptor T and NK cells, and these are the two antigens, PD2R and THSC7A. We know that the majority of autoantibodies for both autoantigens binds in the most N-terminal region of these proteins. So we generated a set of CARs involving the N-terminal region of PD2R and the N-terminal region of THSC7A. And we generated hybridoma cell lines producing antibodies against PD2R and THSC7A. You can see here that they bind nicely to their target in ELISA, and that these hybridoma cell lines also express a B cell receptor, so a surface immunoglobulin. So these are basically the tools that we are using. We also show that these CAR T and CAR NK cells, they express the CAR by staining them with these monoclonal antibodies. And then we use these CAR T and NK cells for both PD2R and THSC7A in vitro experiments to eliminate these hybridoma cell lines. And we always use the other cell line as a control target cell line. So when we test PD2R, we use THSC7A as a control and vice versa. And we see that, yes, both the CAR NK and the CAR T cells are able to eliminate the target cells in an antigen-specific way. So the second approach is the bispecific T-cell engager. You see the principle here again on the left. And we designed constructs involving the cysteine-rich domain of PD2R on the right and an anti-human CD3 nanobody in this case. And we also add a His tag to this construct, so we are able to stain it in FACS analysis and also to purify it actually from cell culture supernatant. This is how it looks in the model, this bispecific T-cell engager in AlphaFold model. And we can express and purify it and see that it's quite clean on a Kumasi gel. Now, this is the BiTE binding to the anti-PD2R hybridoma. On the left is you see that it binds, and on the right is the control using an isotype control monoclonal antibody. So yes, this BiTE binds to the B cell receptor, and then we also validate the binding to a primary human T cell, and you again see the binding on the left, and you see that the negative control is negative here on the right. And then we performed again killing experiments using these target cell lines, using the BiTEs, and using primary human T cells in a fixed effector to target ratio using different concentrations of the BiTE, plus we use control hybridoma cell lines, and we use omitting the T cells as additional control. And yes, we get a very efficient dose-dependent killing of the target cells. And sorry for that. We also do the same experiment with a fixed concentration of the BiTE, but with different effector to target ratios. And again, we see a dose-dependent effect here. So this is a principle that may work in theory, but these are, of course, only in vitro, very preliminary experiments, and we have to see how this may work out in vivo. There's actually one study from Italy from last year, which used a human CD3 transgenic mouse line, immunizing it with PD2R, and then treating it with such a antigen-specific bispecific T-cell engager, showing that the anti-PD2R levels decrease upon treatment with such an engager. However, these experiments, I have to say, are very tricky because it's difficult to distinguish what is actually a depletion of the target cell line and subsequently a drop in antibody levels, and what is just blocking of the circulating antibody because it can bind to the bispecific T-cell engager. And these experiments have to be very carefully controlled. So in summary, basically, for antigen-specific targeting, we have, for example, CAR T cells, and you have heard about the advantages and disadvantages of these compounds. They are usually autologous, they require individualized manufacturing, they are expensive, they may be toxic, and they may generate memory CAR T cells. And I think it's questionable whether this is something that we want in the context of autoimmune disease, which are usually less deadly than cancers where these cells are normally used. We have CAR NK cells. They can be heterologous or allogenic. One donor can be used to treat many patients. They can be used off the shelf. They have less toxicity, and they usually die within a couple of weeks if they are not modified further. And then we have bispecific engagers, which are biologicals. The costs, so yeah, if I produce them, this is a matter of cents. And yeah, of course, this stands in contrast with the whole complex procedure of the autologous manufacturing of CAR T cells. I just want to point out that sometimes the prices for these compounds, yeah, you may argue that they are maybe not rightful in the level they are. They have some toxicity, but probably less than autologous CAR T cells. And the disadvantage may be that it requires, of course, the presence of endogenous T cells. And if you talk about deep B cell depletion in different organs, then yeah, it depends on the presence of effector cells in these organs. Thank you very much.